RT-PCR RNA amplification. Overview. RT-PCR RNA amplification will be considered under the following headings. Introduction. What is PCR? Reaction mixture. Step 1. Denaturation. Step 2. Annealing. Step 3. Extension. Thermal cycles. What is RT-PCR? And reaction mixture. Introduction. RT-PCR is the most widely used test to detect COVID-19 infection. This test involves 4 steps: sample collection, RNA extraction, RNA amplification, and RNA quantification. In the previous video, we discussed about RNA extraction. In this video, we will be reviewing the process of RNA amplification. What is PCR? Polymerase chain reaction or PCR is a technique of DNA amplification. Polymerase refers to an enzyme that forms DNA polymers. Chain reaction is a chemical reaction which progresses in an exponential way. PCR machine is also known as thermal cycler. It involves sequentially changing the temperature of the mixture, known as thermal cycle, in order to produce multiple copies of DNA. Each thermal cycle involves 3 steps: denaturation, annealing, and extension. Reaction mixture. Inside the PCR machine, we have these small tubes in which chemicals are added and reactions take place. Key ingredients of the mixture are DNA template, nucleotides or DNTPs, primers, and Taq polymerase enzyme. DNA template is the segment of original DNA which we want to amplify. Nucleotides or DNTPs are the basic building blocks used for DNA synthesis. They include deoxyadenine triphosphate, deoxycytosine triphosphate, deoxyguanine triphosphate, and deoxythymine triphosphate. Primers are short sequences of nucleotides that help to select the exact portion of DNA that is to be amplified. They are very specific for the DNA template. There are 2 types of primers, the forward primers and the reverse primers. Taq polymerase is an example of heat-resistant DNA polymerase which is frequently used in PCR. As PCR involves thermal cycles and heating, this enzyme remains stable even at higher temperatures. It is named after the bacteria Thermus aquaticus, from which it was isolated. Step 1. Denaturation. For denaturation, machine's temperature is raised to between 94 to 98 degrees Celsius for about 20 to 30 seconds. This causes the hydrogen bonds between the 2 strands of DNA to break and separates 2 strands of DNA template. The temperature and duration depends on the GC content of the sequence, as guanine and cytosine bind together with 3 hydrogen bonds, while thymine and adenine bind together with 2 hydrogen bonds. Step 2. Annealing. During annealing, temperature of machine is brought down to between 50 to 65 degrees Celsius for 20 to 40 seconds. This allows primers to bind their target sequence on the single-stranded DNA. The primers are usually between 18 and 30 nucleotides long. These primers bind to the DNA just next to the sequence of interest on the 3' side. This is because the DNA synthesis takes place from 5' to 3' side. Forward primer is for one strand and reverse primer is for the complementary strand. The temperature depends on the composition of the primer. Higher temperatures hamper with the primer binding to the DNA, while lower temperatures allow primer to bind non-specifically to the DNA. Step 3. Extension. In this step, copying of DNA starts. This step requires a temperature between 75 and 80 degrees Celsius for about 2 minutes. For this step, we need DNA polymerase and nucleotides. Here, the primers provide the hydroxyl group present on the 3' end of the primer that is needed to start the DNA synthesis in 5' to 3' direction. This will allow the polymerase to build a new copy of DNA. At the end of extension step, we would get 2 separate copies of a particular sequence of DNA. Time of 2 minutes is variable and depends on the length of the DNA sequence of interest. Thermal cycles. We can have as many thermal cycles as we wish. After each thermal cycle, the number of DNA sequences of interest double. Therefore, number of copies obtained can be defined as 2 to the power n, where n is the number of thermal cycles. If each thermal cycle takes about 3 minutes and we run PCR for 20 thermal cycles in 60 minutes, then number of copies produced would be 2 to the power 20, which is about 1 million. And if PCR is run for 30 cycles, which might take about 90 minutes, then number of copies produced would be 2 to the power 30, which is about 1 billion. This way, PCR can be used to quickly amplify extremely small amount of DNA sample. What is RT-PCR? As we learned earlier, PCR is a technique of DNA amplification. But the genome of coronavirus consists of single-stranded RNA. To detect the RNA of these viruses with PCR, RNA molecules are first converted with the help of reverse transcriptase enzyme into their complementary single-stranded DNA sequences. Next, the single-stranded DNA is converted into double-stranded DNA by DNA polymerase. Then, the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as reverse transcriptase polymerase chain reaction or RT-PCR. Reaction mixture. RT-PCR is used for detection of the SARS-CoV-2 virus. As discussed earlier, the reaction mixture for PCR contains DNA template, nucleotides, primers, and tag polymerase. The reaction mixture of RT-PCR has the following changes. In place of DNA template, the extracted RNA is added. Here, aim is to amplify the target sequences in the RDRP gene, the E gene, and the N gene. The choice of the target gene depends on the primers and the probe sequences. In addition, the reverse transcriptase enzyme, which converts RNA into complementary DNA, and Tachman probe are added. We will discuss more about Tachman probe in our next video on RNA quantification. Thank you.